I'm here in Denver, Colorado, and it's like 0 degrees outside. So it's the perfect time to talk about some of these um, projects that I've started with mycorrhizal fungi. So just to start off, you can see behind me, there's two little trays, and those trays have morel mushrooms growing in them. I'll get to that later, and I just wanted to start off by doing some updates. We've got our agrocybe parasitica. So we've been growing this one for about 6 weeks now, and it's on bulk substrate. I also isolated a bunch of different phenotypes. So this was the spore streak plate, and then I isolated those colonies from this mushroom, which is from New Zealand. So give us a thumbs up if you can hear me. I'm just waiting for some more people to join in. All right, guys, welcome to Fungi Friday. So I'm just going through some of these plates from last week that I transferred. And if you want to go back on our YouTube video, Fungi Friday number 11, I believe, is the one where we did the spore streaks from the New Zealand mushrooms. And then last week we did a streak plate with Morel mushrooms. So these are those plates after 1 week. And then I made those transfers on Tuesday. So we've got all of these Morel, different kinds of Morels that are growing on agar. So those were the Morchella conica. So these are burn Morels. Please give us a thumbs up if you can hear me. I'm just trying a new setup today. I've got some Morel mushrooms. Cool. All right. All right, guys. So I'm just going over some of the Morel transfers from last week. So I've got my burn Morel, and the Morchella conica. And then you can see the blonde Morel is a lot slower growth, you know, some thicker mycelium. So, and then this one, it looks like there's a little bit of yeast contamination, but that should be okay. So, and then we've got the Morchella importuna. So this one is one of the more versatile morels. It's gonna grow in like wood chips, and it's more saprophytic than most of the other species. But you can see this one is like a really clean plate from the charcoal agar, and that is how fast morel mushrooms can grow. So this is after 4 days growth. It's already covering the plate. And that's kind of the recap from last week. So we also are doing our cordyceps project. I wanted to update on that. So this is the terrestrial fungi. So what's up, Lee? Spores for mushrooms that I've grown, yep. So these are morel mushrooms. And if you check out our YouTube video, we do a Fungi Friday, and last week we isolated some spores on DRBC, so we're gonna start breeding them. And then this is the updated cordyceps, and it's a terrestrial fungi strain, so it's 3 weeks in, nice and orange, and the rest of them are looking like this, so probably about 3 weeks before they start to pin, but I wanted to point out the difference between the United Kingdom. So this is the England cordyceps on agar, and then this is the terrestrial fungi on agar. And you can see these really crazy ridges in there. And I thought that was a really cool morphology. All right, so that's kind of the updates from the projects that we're doing. Now I wanted to talk about the mycorrhizal species. So what are mycorrhizal fungi? Mycorrhizal mushrooms are mushrooms that require trees to grow. So they form a symbiotic relationship or even a parasitic relationship in some cases, but essentially they're exchanging nutrients in the soil by breaking it down so the plants could take it in return they're taking nutrients, which could be byproducts or exudates from the plants, and then the mushrooms are going to use those nutrients. So it kind of requires more layers of complexity to cultivate these mushrooms. So, my belief is that anything that can happen in nature can be replicated in the lab, which is why I've been studying these processes for quite a while. If you can look back on my YouTube channel, I've got a couple videos on my Morel patches that I've been trying to grow for a couple years, and I'm going to be adjusting some of those procedures this fall. So the first problem I'll cover in those past procedures is that I didn't do at the proper time of year. So ideally you want to plant your morels in the fall so that they can freeze. So there's a lot more complexity to growing these types of mushrooms. All right, so the first topic, we'll just get into it, is the morel mushrooms. So you can check out this paper here. It's called Artificial Cultivation of True Morels, Current State, Issues, and Perspectives. And I don't want to try to say these names, but I wanted to give credit to the authors of these papers because they did a lot of the research. I just read it, and I think it's really interesting. So I'm trying to get it out there to try and inspire more people to be growing these types of mushrooms. And it's really fascinating. This paper came out in June 2017, so it's a little dated, but there's a lot of good information. So the first introduction basically covers, what a morel is, the value of morels. There's a huge market for morel mushrooms. They're one of my favorite. They taste really earthy, and if you haven't seen a morel before, it kind of looks like a sponge, like a brown sponge, and they soak up a lot of flavors and are really good in a lot of dishes. So, it would be really cool to be able to cultivate these in the garden or in your yard or even indoors, which I'm attempting right here. So this is my, third or fourth attempt at cultivating morels indoors, and it seems to always stall after it's colonized, and the triggering of sclerotia or conidia is very important, and it's hard to replicate that in the lab. So the life cycle of morels, it's a little bit different than your typical mushroom. So morels are a ascomycete, which it's different from a basidiomycete, which are the typical button mushrooms or all the other mushrooms that I cultivate, except for cordyceps. So it's closer to a cordyceps mushroom than to an oyster mushroom. So they reproduce a little bit differently. They don't just have gills and drop spores. So they have asci, and then those have multiple spores attached to them, and that's what germinates and forms hyphae. So morels are ascomycete. Another interesting feature about morels is that they form sclerotia, or little hard nodules of mycelium. So that's important because over the winter or with big fluctuations in temperature, the sclerotia are going to be a reservoir for nutrients and protect the mycelium and keep them alive. So one confusing thing about cultivating morels is that the spawn looks really different compared to a lot of other mushrooms. So you can see right here is a good example of the sclerotia starting to form, and you can see that the spawn will turn like a dark brown compared to, you know, a typical mushroom that's going to look really white, like this chanterelle here. So this is another mycorrhizal species, and you can see how clumped and amorphic the mycelium is. So that's a really typical property of the mycorrhizal species. So sclerotia, very important. They're not going to fruit unless they have sclerotia. So the next different attribute of morel mushrooms is going to be the formation of conidia. So conidia is a separate part of the growth cycle. They don't really know why it's formed, but you can see on this paper here, there's a pretty good diagram, and this is the morel life cycle. So it's gonna release its spores or its asci, and then it's gonna germinate, and then it forms the mycelium. And in between going from a fruiting body from the mycelium, it's gonna form these conidia. And it's almost like a powdery substance on the surface. That's what it looks like from, you know, a person from up above the soil, but you know, below it's just going to be parts of the mycelium that grow up, and then they form these little pieces that will break off in the wind, and it's like a powdery mildew. So that's also an important part of the morel mushroom, that other mushrooms don't typically do that. And you can see in this example here, if you look very carefully, there's a little bit of conidia starting to form on the surface here. So for making a tray culture, the trick is timing. So this one went a little bit too long, and now it's like covered in mycelium, but I'm still gonna run through with this one because I feel like it's a really strong variety. And you can see very carefully here, there's mycelium that's creeping up into that spawn jar. So one of the most important breakthroughs of morel cultivation is the addition of exogenous grains or nutrients. So you can see in this paper, when they're cultivating the morels, they often put these bags in between the rows, and these bags are filled with nutrients. And the idea is that the mycelium, which is very fast and thin, is constantly looking for nutrients. In nature, it's gonna find an old tree stump, something away from the roots that it can draw its, you know, food from. So I'm not exactly sure who got credit for that, but it's outlined in this paper, and it was one of the most important discoveries because that is what triggers the conidia formation. So after spawning your substrate, you have to wait a couple days until the mycelium start to spread out, and then you want to put your nutrient source in the middle of that substrate, whether it's a jar or a small bag of grains, or in the fields, they just use these bags right here, and the mycelium are gonna search out those bags, start to grab the nutrients, and then they'll start to form the conidia. So if you time that properly, it should happen within the first 2 or 3 weeks from when you spawn your morel spawn. And then you're gonna wanna cover it with some duff, or if you're using a tote, you can just cover it to keep the moisture content up. And then the next important part of morels is the freezing step. So that is kind of, um, so that's kind of a question that is still trying to be answered, is like, how long do they have to freeze for? Can you do this in the lab, or does it have to be outside with other bacteria? So there's a lot of questions, and then they just haven't figured out the complete life cycle of why these conidia form. So someone asked about where I got this paper from. So if you search artificial cultivation of true morels, the current state, issues, and perspectives, it's in the Journal of Critical Reviews in Biotechnology, and it's public, so you can just download this text for free. But essentially, that is the biggest question in cultivating morels: is what do you do after you get the conidia? And if you do it outside, it's kind of up to Mother Nature, which is why it's important to plant them in the fall, because if you do it in the summer, there's a greater chance that the mycelium are going to dry out, and then you're not going to get your fruits. But so moving on from the whole process, there's another important factor. So there's tons and tons of different species of morels, and it's very important to choose a morel that's local to your area, or one that is very adaptive at least. So the types of morels that are outlined in that paper are the Morchella esculata by Groves and Hoare or Hoare. So they did a study on the Morchella esculata, which those are the blonde morels, which is why I chose that species. So in my experience, the mycelium is a lot more fluffy, and it's slower growing. So maybe it's just a stronger strain that has better results. The three other morels that have been successfully cultivated and outlined in this packet are the Importuna, which is a very versatile morel. It can grow in wood chips, the Sextulata, and the Eximia from China. They've also grown Morel conica, which those are burn morels. So there's probably some kind of a substrate additive to induce fruiting from the burn morels. But an interesting note on those is that they only come up after forest fires, so they're probably living in the roots of the trees until there's an event that gets triggered, and then those will fruit. So those might be a little bit harder to fruit unless you can find a really sensitive variety, and then maybe you can cultivate those. All right, so that will cover the Morchella, the different species. So another thing to consider about the morel mushrooms is they're more trophic than other mycorrhizal species, which I believe that's why they're the first that have been cultivated. So they form a symbiotic relationship with trees, which is mycorrhizal, but they also can have relationships with orchids, which is very convoluted, and also it can live in the trees and not be triggered until a forest fire. So it's a very elusive mycelium. It can jump from different trees to plants, and there's some studies that it's saprophytic in nature too, so it can feed on dead trees. So that's why it's so amorphic, and I believe that that's the first mycorrhizal species that can be cultivated. There's a lot of promise. They're doing this commercially in China. I think there might be a couple farms in America now, but if someone can figure out how to do it indoor, and sustain it, then that's going to be a really valuable process. So that being said, there have been a few commercial morel farms in the 80s and the 90s, And lately there was one in the early 2000s, but typically the problem with those is that the strain senesces so quickly with Morchella. I think because it's the ascomycete, just like cordyceps mushrooms. It's hard to maintain the culture. So if you were to produce it indoors, it's important to keep a mother culture that's, cryo-frozen or something that you can go back to easily, or else you're going to have to constantly be breeding new strains so that they won't break down over time. All right. So that kind of covers the morel mushrooms. And now I would like to move on to another project that I'm doing that I'm really excited about. And it's the cultivation of Boletus edulis. So my favorite mushroom is the porcini, and you can check out this paper. It's called the mycorrhization between Cistus ladanifer and Boletus edulis. And it's enhanced by the mycorrhizae helper Pseudomonas fluorescens. So this is a paper that came out of Spain. It's on the Mycorrhiza Journal from 2016, so it's public. And it talks all about the relationship between porcini mushrooms, this bush, Cistus ladanifer, which is a really cool bush, and then also it incorporates Pseudomonas fluorescens, which is a really cool bacteria. So there's 3 different kingdoms working together to produce these mushrooms. And it just starts off by describing the relationship between Boletus edulis and plants. So Boletus edulis, it's a mycorrhizal mushroom. We have some of this on agar, and it's also on a antibiotic agar because I wanted to isolate it from bacteria. So I have a couple different phenotypes. One of them is really aggressive and it can grow just as quickly as the morel mushrooms. And this is what it looks like on liquid culture. So one of the downsides to this one is it's very slow growing, but I put it into a liquid culture so that I could quickly expand it out onto grains. And then my plan is in the springtime, I'm going to plant it outside with these special bushes and then drench it with the bacteria. So, Cistus ladanifer. This is a really cool bush. It's native to Spain, but there are other species that are very similar. It's a rock rose, that's the common name, and it's like a white flower with little black spots. Super cool. Luckily, I found a strain that can withstand the cold winters here in Colorado. It's still going to be sensitive, so I'm going to have to cover it, but this bush is a really cool fungi reservoir. So the Cistus ladanifer, it only appears after forest fires, and it's been shown to harbor over 400 different species of fungi. And the theory behind this is that it's one of the first plants to pop up after a forest fire. So all of the mycorrhizal mushrooms that would be burned down from the trees and die jump onto this plant and kind of like harbor themselves there until the forest starts to regrow. So it's a very adaptable bush as far as, mushrooms and bacteria go. It has some characteristics that the Boletus edulis really likes. It kind of gets into the morphology in this paper, but basically there's a few different types of mycorrhizal mushrooms. You can have an endomycorrhizal mushroom that goes inside the roots of the tree and forms a connection, or an ectomycorrhizal relationship, and it kind of forms a web or like a net right around the edges of the roots of either a tree or, in this case, the bush. So the porcini is an ectomycorrhizal mushroom. Also chanterelles, which I've got some chanterelles in a liquid culture, and I'm also going to be attempting this same procedure with a chanterelle. There's not too much studies on that, but I feel like they're very similar. So, all right. So the Boletus edulis has to form this relationship with a specific tree. The tree has to allow for the mushroom to inhabit, the roots underneath the soil. In order for this to happen, there's a specific bacteria that helps bridge the gap between the hyphae and the roots, the meristems. So this bacteria is called Pseudomonas fluorescens, and you can see I got it in a little vial. It's not a harmful bacteria. It's everywhere in the soil. It's pretty ubiquitous in nature. They call it Pseudomonas fluorescens because it fluoresces under UV, so it glows in the dark. It's really cool. It has flagella on the exterior. It's a gram-negative bacillus, which means it's kind of shaped like a rod, and then it has these fuzzy flagella, which allow it to move around easily in the soil. So another feature of this bacteria is that it bridges the gap between the hyphae and the roots of the tree, or in this case, a bush. I think it's one of the coolest bacteria out there, and this whole paper goes into detail the mechanisms of how it works, but essentially they did a study where they planted mycelium with the bacteria, mycelium without the bacteria, and there's a lot of regional data on the climate, the weather, so it's very specific to a region, but they found out that the plants that contained the bacteria and the mycelium, those mycorrhizal connections lasted months and months and months longer than the mycelium that didn't. There was about a 30% success, maybe a little bit more, when they inoculated this spawn with the bush and then they moved it out into the fields. So the idea of this whole paper was that in the nurseries they can inoculate plants with the mycorrhizal fungi and water it in with the bacteria, and then they can use those to kind of bring back some of these old farmland that's kind of dead from previous farming. So they're trying to bring back the life and the ecosystem by using the mushrooms. However, I think you can just kind of plant them in rows and use the same principles and then get Boletus edulis. So they're really cool bush to look at. It's just a really pretty plant, and it's healthy for the soil. There's some studies that they could spread up to 30 meters away, and I think that the reason for this is because after a forest fire, the mycorrhizal mushroom is really stressed out and it has to grab onto anything possible. So if a brand new tree is starting 30 meters away, it's gonna stretch out and inhabit that tree until it becomes an adult, and then the whole cycle will start all over. So really, really cool study, and it's from the Mycorrhizae journal, and I don't want to try to pronounce these people's names, but definitely an interesting article. So then that takes us up to November 2020, and there's this article about the use of inoculator bacteria to promote tuber melanosporum. So that's a truffle mushroom. So this is a brand new study. It came out a couple months ago, and it describes the same procedure with the Pseudomonas fluorescens that they used from this Boletus edulis, and they use it on some tuber mushrooms, or some truffles, and they found a lot of, you know, promising results as well. The addition of the bacteria really helps in the life cycle of the tuber. So I feel like there's, you know, a relationship between these three kingdoms that is very fragile, and it's important to consider when you're trying to grow these mushrooms. So that kind of, you know, concludes my overview of these different articles and kind of my plan moving forward. So if you guys want to follow me on YouTube, I'm going to be continuing this series on the Morel cultivation. I'm going to be doing a spring project with the Boletus Edulis, as well as chanterelle mushrooms. So I'm going to be planting these guys in a similar way, just kind of off near my oak tree. And then I'm going to be drenching with this Pseudomonas fluorescens. So in the springtime, I'll probably do 2 different drenches. It describes, you know, the concentration in these papers. So I'm just gonna use those as a reference and kind of consider, you know, how it's going to play out. And then hopefully in the springtime, I'll get some fruits from my last year Morel, or I'll be planting these Morels again this fall. And then I also have a whole project planned for how to utilize mushrooms in the garden. So I've got some King Stropharia strains that I've been growing out. I'm gonna be expanding those shortly. And then maybe in early March, I'm going to build some raised beds and then incorporate mushrooms in those beds, as well as capture some of the humidity from my grow tents now. So it's gonna create a high CO2 environment for the plants, as well as a high humidity environment to cut down on watering. And it's going to, you know, correlate with some of these mycorrhizal mushrooms that I'm gonna be planting in the springtime. So stay tuned. Give us a thumbs up if you enjoyed this video. Check out our Etsy. We're gonna be having, you know, more of these liquid cultures available on Etsy, and possibly in the springtime, I'm gonna sell ready to grow bags of the Stropharia, as well as in the fall, I want to have spawn for Morel mushrooms available. So check out our Etsy, it's Fresh Fungi. And then tune in next week, Friday at 4 p.m. for Fungi Friday number 16. Haven't figured it out yet, but I'll just give you guys updates on all these projects, and I'll be releasing smaller videos throughout the weeks. So thanks for joining in. I appreciate you guys following along, and if you have any comments, check out our YouTube video, and I'm really excited to hear some suggestions, and if anyone else has resources out there on how to, you know, contribute to this project, feel free to comment. Until next time, much love.